The next speaker will be John Kubie.
Great seeing you all in person. It's been a difficult 2 years, lots of teaching remotely and other things remotely. It just doesn't, as we're finding, it doesn't work. And I'm really happy to see live faces, and I think we all appreciate the difference. The title of this talk, I think, is Hippocampus and Navigation, which on its surface does not probably sound like learning and memory, which is the symposium. I'm going to talk mostly about the hippocampus and navigation and play cells. And at the end, hopefully, present how it's related to learning and memory. This talk should have come before Andre's talk. It's much, much more basic, and it's really largely historical about what's going on in play cell research at Downstate. Now, there have been a few, I think, revolutions in neuroscience related to the hippocampus. Early on, say up through the 1960s, the correct strategy in studying the nervous system was to go from the periphery inward, from sensory receptors, step by step by step, or from motor systems back into the brain. And the idea of understanding what's going on in the middle of the brain seemed outlandish and wrong. It seemed like it couldn't work. Now, the revolutions in hippocampus, one of them is not talked about here. It's Scoville and Milner's discovery that hippocampal lesions in humans produced profound amnesia. The paper was 1957. Before that, it was sort of thought the hippocampus was redundant and didn't really need it. So that's revolution number one, still being heard today. Revolution number two is illustrated here, and started a paper by John O'Keefe and Ostrovsky, where he described hippocampal play cells, and I'll describe them a little bit more fully later. Andre has already given you some more complicated examples. And I think equally importantly, in 1978, O'Keefe and his collaborator Lynn Nadell wrote a really remarkable book, The Hippocampus, a Cognitive Map, which laid out this really crazy, bold idea, which reformulated, based on very little, maybe half a dozen play cells that O'Keefe had recorded, but it laid out this framework that, first of all, that hippocampus was a mapping system, but probably most important, that it was a cognitive mapping system. They were approaching cognition, which really had never been approached before, I believe, at a neuronal basis. They were laying out a scheme for exploring cognition in neurons in the brain. And although this was done in 1978, it's still the point of reference we all use, although most of this is sort of wrong. It's still the basic framework that the hippocampus is a cognitive system, and one of its fundamental roles is in mapping. Okay, so how do I go forward? How was that? Okay, so that was O'Keefe discovered this in the early 70s. Jim Ronk moved to Downstate in 1975. This picture's not here, and since roughly that time through close to the present, a whole bunch of people have come through that group. It's here, a bunch of faces. Some are familiar, like Andre and Mark Stewart and me and Bob Muller. Some of you may hopefully remember, and Steve Fox. These pictures were chosen not in terms of seniority or importance, but in terms of people whose pictures I could find. But most important, I think, trying to go forward. Most important, Jim was the catalyst. He came in 1975 and was a terrific center of gravity throughout. Really wonderful guy, for people who know him. Okay, so what's a play cell? I'm not hoping you can hear sound. This is recording a single neuron. Illustrated over here is a kind of electrode, fine wires going into the hippocampus. And single electrodes are close enough to neurons that they can record the action potentials. I think you can hear the action potentials as sound waves. And so this is recording a single neuron from this rat's brain. The rat's not particularly bothered by it. The light bulbs are so we can know where the rat is. And if you record for 15 or so minutes, you create a map. And the color code here represents pixels on the floor of this apparatus. Yellow pixels are regions the rat visited where there were 0 spikes. And the darker colors up to purple are higher firing rates, averaged over the 15 minutes, with the purple ones being about 18 spikes per second. So really a fairly dramatic signal to measure effect. So this is an example of a single flash. You can insert here a second cell smaller over here. Sometimes you can get two cells in a single electrode. This dates back to the 1980s, but I hope you can get a sense of what a place cell is. There it's firing again. And they are behavioral correlatives. The animal is freely moving in its two-dimensional apparatus, actually chasing food pellets randomly thrown to the floor. It's a task that doesn't require the hippocampus, but the cell's firing station. And that came from our earliest studies, one of our earliest studies. And here's a sampler of different place cells. Notion is they could be anywhere in this apparatus, roughly equally distributed, not always the same shape, fairly large in shape. Here is a cell that was recorded that has very, very few spikes. I think maybe about 20 in a 15-minute session. And this is a place cell because when we took the rat and this cell to a different apparatus, it had a robust firing field, which is tricky but important finding. So Andre mentioned this earlier. Now, knowing what single place cells are, you can well imagine, as we did, that if you were able to report from tens or hundreds or thousands, they would distribute across the entirety of the floor. And that was O'Keefe's idea, that it formed a map. So what's the value of a map? The map for the rat can do two things. One, it can tell the animal where it is in location, self-localization. The other is that it can help it navigate. And there's evidence for both. The navigational evidence is that rats with hippocampal damage basically are very, very poor at navigating. Navigating isn't always by a map-like system. They can go to landmarks, but they can't use what's considered a map-like strategy. So those are the two functions of these, the presumed functions of these hippocampal neurons. They're tell the GPS system, tell the animal where it is, and a navigational system where it can go efficiently. Now, getting back to a broader context, there are roughly three big areas or three seemingly separate areas in hippocampal neuroscience. One is what I talked about earlier, the Scoville and Milner finding of amnesia, hippocampal damage. Second is hippocampal place cells, and here is illustrated a water maze, which is the converse. That is, there's damage to the hippocampus. The animals can't navigate well. And the third, which has been talked about by Todd and Juan Marcos, is recording synaptic plasticity in the hippocampus, which talks to memory, but it's not that based on circuitry, how it deals with the brain. And so the last slide I have is sort of a very, very quick summary. I don't expect you to read it all, of some of the contributions that the group at Downstate has made over the 30 or so years. And a couple of them, we were really the first to make these computer-based data collection and maps, which I think was fundamental in convincing people that O'Keefe was crazy. It became a standard for how you really look at these things. And another thing which I sort of indirectly pointed to is the idea of remapping. That is, when you take an animal from one environment to another, the place cells scatter randomly. Initially, we thought that's crazy. If you want to map, they should all sort of keep relationships, but they don't. The opposite happens. They scatter randomly. And now we think that that's what a second function of the hippocampus, which is context. It doesn't only tell the animal exactly its location, but in this case, the context is I'm in this room at SUNY Global Center, and what are the possible things to do here? Zero time? 10 was okay. So, and I have to mention that Jim Ronk discovered head direction cells. So how does this get to memory? And there's some background findings that are real important that weren't downstate findings. And that is when you have a series of place cells, when the animal goes along a path, you can imagine this, a chain of place cell firing. And we'll call that a path, a firing path. Well, that firing path does not only occur when the animal's walking, it recurs other times. And importantly, it recurs during the next sleeping epoch. So if an animal walks through a chamber and has maybe 50 discrete paths, there are 50 possible epochs. And we can think of those as episodes, or the cellular basis of an episodic event. And these episodes replay during sleep. these episodic sequences. And it's been now very clearly shown that the replay of these episodic sequences is critical for the consolidation of episodic memories. That's it.